Good evening, and welcome to the Marion Minor Cook Athenaeum. My name is Michael Grauske, and I am one of the Ath Fellows this year. In the earliest stages of the HIV-AIDS pandemic, the public health battle was as much against fear as it was against the disease itself. Throughout much of the 1980s, uncertainty about AIDS effects and the ways in which it could be transmitted from one person to another led to widespread panic. While today HIV-AIDS remains a serious and often stigmatized condition, our improved understanding of the disease and the development of antiretroviral drugs has reduced much of the fear and anxiety experienced by previous generations. Tonight's speaker, Karl Haushalter, is a professor of chemistry and biology at Harvey-Mudd College, who will share insights into the development of antiretroviral drugs, which have transformed an HIV-AIDS infection from a deadly illness into a manageable condition. In addition, Professor Haushalter will discuss the latest advances in antiretroviral therapy and the hopes for a cure for HIV. Professor Haushalter holds a BA in chemistry from Rice University and a PhD in chemical biology from Harvard University. He has been a member of the Harvey-Mudd faculty since 2003, where he is also the Associate Dean of Research and Experiential Learning. Professor Haushalter serves on the board of directors of Foothill AIDS Project and holds an adjunct faculty appointment at the City of Hope National Medical Center. As always, audio and visual recording is prohibited. Please join me in welcoming Professor Haushalter to the Athenaeum.
Good evening, and thank you so much to the students for coming out on it, which I'm sure is a very busy season in the academics. I'm really glad to see so many people here, see a lot of familiar faces. And so thank you so much for coming out and hearing this talk. Thank you, Michael, for that introduction. I've attended a number of Ath events and always really enjoyed being a participant, so it feels very much like an honor to be up here on this side of the presentation. I'm a very hopeful person, and so tonight, I'll share with you my hopes with regards to HIV. As Michael said, I'm a professor at Harvey-Mudd College. It's my day job. I love it there. One of the things that I do in addition to my work at Harvey-Mudd is work with Foothill AIDS Project. And the stories that I'm going to tell you tonight, what I've learned about HIV, is as much what I've learned in the lab as what I've learned from working with this organization that's based right here in Claremont. So HIV, I think we could accurately say it's forgotten, but not gone. This is a heat map showing the incidence of HIV in Los Angeles County broken down by census tracts. And you can see that there's these very concentrated pockets that correlate really well with poverty here in Los Angeles County. Some numbers, there's about 60,000 people currently living with HIV here in LA County. The median age of those living with HIV is currently 48. That's up from 41 in 2002. And the group with the highest rate of new infections are young people. So there's a few things going on. This number, 60,000, is going up, and it's going up every year. And it's going up for a good reason and for a bad reason. The good reason is that people are living longer. People who have HIV are able to take a medicine, a pill. For many of them, it's just a single pill per day that enables them to live with the virus for decades. And so people are living a lot longer. The HIV positive population is growing. So that's the good news. The bad news is that there's an alarmingly high rate of new infections, especially among young people, especially among young gay men of color, that is contributing to an increase in new infections. And so we're going to talk a little bit tonight about both of these, like the progress that's been made in treatment and what we still have to do in order to address the ongoing rates of transmission. So with regards to treatment, what we're looking at now is a plot of the mortality due to HIV in the United States with time. And you can see that it was going up and up and up, and peaked in the mid-90s. And actually, in the mid-90s, HIV was the leading cause of death for adults in America aged 24 to 41. And so something happened there. Something happened for this dramatic fall. And what happened was the development of a protocol to treat people with HIV in which they would take not one pill, not two pills, but three separate pills, the triple cocktail. And this combination of potent antiretroviral medicines would essentially block HIV replication, stop the virus from progressing, and restore immune function. And this triple cocktail therapy then has enabled people's lives to be restored. Without treatment, someone who newly acquires HIV has a rapid rise in their viral load. The immune system kicks in a few short weeks after that and is able to hold the virus at bay for months, years, decade maybe, but eventually the virus takes over. The virus will wear the immune system down, spiral out of control, and it's at this point here that an HIV infection progresses to the condition known as AIDS, where the immune system fails and the body is no longer able to fight off microorganisms and opportunistic infections arise. With highly active antiretroviral therapy, with the triple cocktail, the pattern is very different. The viral load goes up. Once a person goes on highly active antiretroviral therapy, starts taking the pills, viral load goes down, and this dashed line is meant to represent the sensitivity of the clinical assay used to measure viral load. And it's actually below the limit of detection. And so someone can become undetectably low virus load by taking this medicine. So it's a very effective therapy. And since the mid-90s, we've continued to make progress on this. And so the newer therapies are much more potent, that they have fewer and less severe side effects. And instead of having to take this number of pills per day, which is quite challenging, most people newly infected with HIV take this number of pills per day. So we continue to make a lot of improvements in the convenience of this, the toxicity being lowered, and just the overall effectiveness of blocking the virus. So this shows then, starting from mid-1996, how we continue to decrease the mortality due to HIV. This is a study done by Kaiser Permanente of their HIV-positive patients. There still is a gap, but the life expectancy gap for someone living with HIV is somewhere between 8 to 12 years now. It used to be decades, the gap between someone positive and someone negative. That gap is as small as 8 to 12 years. It's getting smaller every year. These two lines are converging. So as we talk about the antiretrovirals, I want to say a little bit about what they do. They block active replication of the virus. They stop the virus from making copies of itself. The virus is very, very proficient. It's just an incredibly high-powered Xerox machine just running ditto copies of itself. The antiretrovirals stop this process completely. They unplug the ditto machine. This then restores most, not all, but most of the function of the immune system, and therefore then significantly extends the lifespan and improves the quality of life. And what's really come out recently, and I'll say more about this at the later part of the talk, is that someone who's on antiretrovirals also is dramatically less likely to pass the virus on to a sexual partner or someone that they might be sharing needles with. And so the antiretrovirals are a huge tool for prevention as well as for treatment. Now, this all sounds good. There's a few things that we should be careful to keep in mind that they don't do. The antiretrovirals do not cure HIV infection. We do not yet have a way to cure HIV infection. And this is because they do not eliminate the virus. They do not kill the virus. They do not eliminate the virus. All they do is stop the virus from making copies of itself. And so the virus is not eliminated by the antiretrovirals. And this may seem so obvious I shouldn't need to say it, but in the real world, this is one of the biggest challenges. They don't work unless the person takes the pills. And why someone might not take this medicine is complex and varied. Some of it has to do with access. Some of it has to do with other life factors. We'll talk more about that as well. So, as a biochemist, I'm incredibly proud that my field was able to make this contribution. of the antiretrovirals. And the way that these drugs were developed was by very carefully understanding the process by which the virus will attach and then fuse with the cell that it infects, make a copy of its genetic material, insert that genetic material into the genome of the cell that it infects, and then from that genomic material make more copies of itself, which then bud off and pinch off. And scientists over the years have very carefully dissected every molecular detail of this step, this very complicated choreography that goes on to produce new virus. We now understand the details of, the exquisite details. And so that's allowed us then to target and block the virus at multiple steps in this pathway, from the initial attachment to the copying step, to the integration step, to a step that's involved in processing that then enables the virus to bud off to make a mature particle. And so the drugs that we have target multiple steps in these pathways. The ones that have, we started with, the earliest drugs were drugs that inhibited an enzyme called reverse transcriptase, that the enzyme that the virus uses to copy its RNA genome into DNA. Later, the protease inhibitors came along. This was in the mid-90s. That was part of the original triple cocktail. Nowadays, in 2016, the drugs that are most commonly used, the most effective, most potent, fewest side effects, are integrase inhibitors. And these are drugs that block the DNA of the virus from being inserted into the DNA of the cell that it infects. And so it's through this really detailed molecular understanding of the events of the virus that we've been able to develop these drugs. Now, this all sounds wonderful, so why have we not cured it? Like, why have we not, given all this technology, all that's been done, why haven't we been able to cure HIV? Why can't we eliminate it? So I'm going to use a little bit of a cartoon to illustrate this. These white blobs here are meant to be helper T cells. So these are the white blood cells that are involved in coordinating your immune system's defending the body from infections. And then here's the virus. When the virus initially infects a new cell, it'll insert a copy of its genome into the genome of the cell that it infects. These little squiggly lines here are meant to represent the genome of the cell. And here's the genome of the cell with this extra bit of DNA in it. And most of the time what happens then is that this cell with the DNA will express the RNA and the proteins from this DNA to then make new viruses, which then pinch off and replenish the pool of virus. And you have this vicious cycle where the newly formed virus then will infect more cells, which will then make more virus, which will then make more cells. And just around and around and around it goes. And something like 10 billion virus particles can be made in a day. It's just an astonishing number. This is what happens most of the time. A fraction of the time, 1 in a million, perhaps, instead of this newly infected cell, which has the DNA from HIV, instead of that DNA being turned on, that DNA remains silent. And so all the instructions are there. The blueprint for making more virus is there, but it's not being operated on. It's not being utilized. These genes are silent. So this silent genome means that we have this silent cell, that it has all the instructions, but is not actually making new virus. And we're going to see just in a moment about why that matters. So the antiretroviral medicines, and so when you take your integrase inhibitor or your protease inhibitor or your reverse transcriptase inhibitor, whatever it is, when you take the ARVs, what these do is they block this vicious cycle. They interrupt this cycle. They form a wall that then prevents the virus from infecting new cells. And so this big wall blocks this cycle. So there are no more newly infected cells because viral replication has been shut down. These actively infected cells don't live that long. It's very stressful on the cell to be making virus, as well as the immune system can recognize HIV and destroy these cells. So these cells have a very short lifetime. In a few weeks, they'll decay and go away. And the virus actually has an even shorter lifetime. It doesn't survive very well at all without making a copy of itself. And so the virus gradually will go away. And then if you give a blood test to someone who's successful with their antiretroviral medicine, the result will be undetectable, an undetectable level of virus. Super. Have we cured the person? A lot of shaking hands. Why not? Right there. So, through a mechanism that's not completely understood, these latently infected cells appear to reactivate at some low but steady rate to become active to then make a little bit of virus. And then that little bit of virus then is not able to further infect other cells. And so this virus is blocked from infecting new cells. And so things don't continue, but it's still there. And so the truth be told about this viral load undetectable, if you go in, not with the assay that's used clinically and commercially, but more sensitive techniques, things like quantitative PCR, you can go and you can find a tiny little bit of virus. And it's always there. It's never zero. It's always just a tiny little bit. And we believe that's from these latently infected cells that become reactivated and then make a little bit of virus. But then with the antiretroviral block, it stops. Okay, so this is fine. But what happens, well, it's a stalemate. And so the medicine has to be kept on continuously. And this is why it's so important that the antiretrovirals are taken for the rest of this person's life on a consistent basis. Because what happens if you don't? If you go on a drug furlough, it used to be called a drug holiday, but I don't like that term because I like holidays, but no one likes a furlough. No one wants to be furloughed. And so if you put your antiretrovirals on furlough, what happens? Well, if that block is gone, the cycle can start up again. And once the cycle starts up again, you can make more virus, and it takes over. And this process happens very quickly. Matter of weeks or so, someone can jump from virus load undetectable to tens of thousands, hundreds of thousands of viral particles per mil of blood. And so that's why it's so important that the treatment is given consistently throughout. Like, very, very important to have this consistency to prevent the viral rebound. So here we are. Now, people have made, as I told you, better and better antiretrovirals. And so the antiretrovirals we use now in 2016 are better than the ones that we used in 1996 when this therapy first came along. They're more potent. So in other words, this wall is now much higher. It's much more effective. We've essentially been able to completely shut down viral replication. And it's easier to take, so people are more successful with it. There's fewer side effects. But making better and better antiretrovirals will just make that blue block even better and better and better. But that's not actually getting at the problem. And so if you wanted to cure someone, you're going to at some point have to deal with the latent cells. And antiretrovirals, which block viral replication, are not going to do it. You need a different approach. And here is, and at this point, this has not been achieved yet. It's something that we're trying to do. There's clinical trials researching this. This is where the new frontier is an approach that's called kick and kill. And we're going to probably need to come up with a better name if it actually ends up going mainstream. But kick and kill is the approach. And so we kick, so using something called a latency reversing agent, we drive these latently infected cells from the off state to the on state. That's the kick part. Now, at first glance, that sounds like a horrible idea because if you do so, what you're doing is you're taking this cell that's causing no harm, just sort of hanging out, and it's now going to be activated to a cell that is making more and more and more virus. But the latency reversing agent is given simultaneous with an antiretroviral cocktail, such that any virus that's made is not able to establish any new infections. And so the hope then is if you give a potent enough kick, and a lot of studies have shown that this is probably going to need to be more than one agent, probably delivered more than once, but if you're able to move all of these cells from this state to this state, they go away. And we already know that these cells eventually die away, and the virus goes away. And then potentially, we can take the antiretrovirals away. And so this has the potential to eliminate the need to continue to take the antiretrovirals every day. This could be curative. And I hope I qualified that enough with enough could-haves and potentials. We have a long ways to go here. We're not very good at kicking yet. Like we haven't figured out how to reactivate the virus effectively. We can get a fraction of those latently infected cells activated. We need, by most estimates, to get almost all of them. Something like, we can miss 1 in a million, maybe, but almost all of them need to be activated. And that's a very tall challenge. So we're gonna need to figure out the drugs to do that. We also need to get better at actually killing the cells that are reactivated. HIV has been able to survive a long time because of its ability to escape the immune system. And so we need to figure out ways to boost the ability of the body to actually kill those cells that are reactivated. And we need to do all of this while addressing really important safety concerns. And so this is promising, but is still probably several years away from actually coming to fruition. So in the meantime, as much as I'm hopeful about our prospects for a cure and what that would mean for people who are HIV positive, we need to be very mindful of where we are in the present and being sure that we're addressing our current needs. And with regards to our current needs, certainly it's treating people who already are HIV positive, but a lot of what we can be doing is to prevent new infections in the first place, because HIV is preventable. And the best preventative approach, much like the best therapeutic approach, is a combination approach that takes into account behavioral changes, biomedical strategies such as male circumcision. Male circumcision reduces the probability of acquiring HIV by 60% for males. And so that's an approach that's shown to be really beneficial. There's a lot of issues regards to social justice and human rights connected to HIV, things like criminalization, and how progressive policies are with regards to injecting drug users, and opioid substitution therapy and needle exchange programs that need to be taken into account. And then also the antiretrovirals themselves can be part of a prevention package, and that's what I'm going to focus on. And so for the next few minutes, I'm going to be talking about using the antiretrovirals for prevention. I just want to make sure that I'm clear that the drugs alone are not sufficient to really have a comprehensive prevention package. It really has to have all these approaches going on in a community simultaneously. I'm going to be focusing on my area of expertise, the antiretrovirals, but just be sure that you keep in mind that what I'm about to tell you is in the context of all these other very important efforts that are also going on. So the thinking goes a little bit like this. Given that the antiretrovirals are able to decrease the viral load of the person taking them, what does that mean about their ability to pass on the virus? And we've thought for a long time that by reducing the viral load, that should, it seems, be reasonable to assume that this would actually mean that the person would be less likely to pass it on to a sexual partner. And we've thought about this for a long time. We've now started to come up with a number of studies to look at this directly, and it turns out to be the case. The people who are virally suppressed are dramatically less likely to pass on the virus. And the probability of transmission from someone who's positive to someone who's negative, if the positive person is on successful therapy, that probability is really, really low. And in fact, there was a group, the Partners study, that tried to figure out how low is it. And so what they did is they took about 1,200 serodiscordant couples. And a serodiscordant couple is a couple where one member of the couple is positive, the other is negative. And the couples recruited into this study were a mixture of heterosexual couples and male-male couples. For all of the couples, one partner was positive, one was negative. and the positive partner was on antiretrovirals and had a viral load that was low and was stably low. And for this particular study, they recruited couples who self-reported to not use condoms consistently. It is the recommendation for shared discordant couples. Not everyone follows that. These were people recruited into the study because they reported that they were not consistent in their condom usage. And so over the course of the study, they did regular follow-ups and asking about sexual behaviors, about condom usage. They were always advised and counseled to use condoms, provided condoms, but they collected data on the activity that was happening. And over the course of the study, they accumulated 36,000 acts from the heterosexual couples and 22,000 sexual acts without condoms with the men who have sex with men couples. So that's 58,000 condomless sexual acts. And these were all acts between someone who was positive and someone who was negative without a condom. So how many new infections do you think there were? 0. None. 58,000 exposures. There were no linked new infections. Now, I need to explain that just a little bit. It wasn't 0 new infections. So there was, in this study, there were 1,200 negative, HIV negative people. There were, at the end of this, 11 newly positive people who were negative at the start of the study, who are now positive. They were not linked. And this is part of what was new about this study, and part of why this study has been so powerful, is because that they were able to sequence the virus of the person who was newly positive, sequence the virus of their partner, sequence the virus in the population, and then make a determination, did the virus of the person who was negative, who became positive, was that virus the same as the virus of their positive partner, or was it more likely to be from someone else? And so the result was that the people who became positive during the course of the study, there was 11 of them, they were all with people other than their primary partner. So that's the word linked. But if talking just about within the couples where the partner was on the med stably, there were 0 linked new infections. And so they haven't found any yet. And the study is still ongoing. And so maybe one will appear, but as of this point, there have been 0 observations. This, by the way, doesn't mean that the probability of transmission is 0. It just means that it's really low. And we don't know how low yet until we do more studies and accumulate more acts so we can get a better handle on how low the probability is. At this point, we can say to the 95% confidence level that the rate is less than 0.3 transmission events per 100 person years. And so that's close to 0. But we can't yet say exactly how close to 0 it is. But it is really phenomenally low. And so this is just showing that there's a false dichotomy between treatment and prevention. Treatment is prevention because successful treatment dramatically eliminates, maybe, but at least dramatically reduces the probability of transmission. The other way that the antiretrovirals have been used for prevention is pre-exposure prophylaxis, or PrEP. And so many people have probably heard of PrEP. PrEP is relatively new, about 5 years or so. And it involves giving a medicine, Truvada, which is actually a combination of 2 of the drugs that are commonly used in the triple cocktail, not to the person who is positive, but to the person who is negative, who anticipates that they might be exposed to someone who is positive, and they would take this drug with the hopes that in doing so they would prevent acquisition. Much the same way that if you were traveling to a part of the world where malaria is endemic, it's common protocol to then take the anti-malarial drugs before you go and while you're there. In the same way, pre-exposure prophylaxis involves taking this medicine prior to and during exposure to prevent the possibility of acquisition. It's believed to work by blocking the very early local replication right at the port of entry before the infection can then go systemic within the body. And clinical trials have shown that it's very effective when it's used. It's not as effective if it's prescribed and not used. It turns out to be not very effective at all in those circumstances. But if you actually take the medicine, it's been shown to reduce the probability of transmission dramatically. And there's a lot of potential roles for using PrEP. An important consideration is that it gives the HIV negative person a sense of agency. Not every sexual relationship involves a power dynamic in which both partners feel equally able to dictate the terms of how the sex will occur. And HIV negative people can take PrEP without their partner consenting or without their partner even knowing or having to know at all. And so it gives that person agency, which I think is an important consideration. It also gives another means of protection for serodiscordant couples. If the partner study that I showed you before isn't enough protection, this gives you yet another layer of protection, and especially important if you're considering having a child, where the use of a condom would, of course, not be a part of that situation. And there's a number of reasons why someone might choose to engage in sex without a condom, having to do with the particular culture that they're a member of that's important to them, intimacy, pleasure, or other reasons. And so I think the reality is many people do not use condoms currently, and so this gives them a protection option. And another important consideration is that PrEP brings the HIV negative person into systems of care. And so this means that they're actually going to be seeing their physician, talking about their sexual health with the physician, which has other positive benefits for the person. Now, there's obviously a lot of questions and concerns about this. It is relatively new. A big one, and this is what a lot of the critics of this approach will argue against very, very stridently about, is that with PrEP, with these pills that prevent HIV, would that disincentivize condom use? Do people engage in risk compensation, where they might have more sexual partners, more sexual acts without a condom because they have this pill? And the main concern being that the pill, although it's effective, is not 100% effective. So there's always a little bit of possible HIV infection going on, as well as HIV is not the only sexually transmitted disease. Here in Los Angeles County, chlamydia is actually one of the most commonly diagnosed sexually transmitted disease, and gonorrhea and syphilis. These are all sexually transmitted disease that condoms provide protection for, but PrEP, this anti-HIV pill, would not. Would people actually take the pills? That was one of the biggest things that came out of the clinical trials, was people who took the pills were protected, people who didn't take the pills were not. What about the long-term side effects of having to take this medicine? Will it lead to an increase of drug resistance? Some people have suggested that perhaps pharmaceutical companies, in particular Gilead, which manufactures Truvada, have been playing an undue role in promoting PrEP. And then there's just a question of resources about, is it worth it? And these sort of get into economics and the politics of HIV. These questions, though, the top four, are questions that we can do clinical research on and get the answers to. And a lot of the community level research that's going on, sort of outside of a clinical trial, like actual implementation, shows that a lot of these concerns are, while very valid concerns, like the worst possible fears haven't emerged. That risk compensation and behavior change has not occurred to the extent which people forecast. That people who were not using condoms previously continue to not use condoms. People who do use condoms continue to use them for the most part. That hasn't particularly changed. That Truvada was chosen because it has the lowest side effect profile of the antiretrovirals on the market. So they are reasonably safe drugs, and we have not been seeing a huge rise of drug-resistant strains in the early years of PrEP. These are, of course, things we'll continue to monitor and to look at, as a public health community. But a lot of the early data from the early experience using PrEP looks very positive. And there's actually a lot of parallels to the advent of oral contraception. The resistance that PrEP has faced is very similar to the resistance to oral contraceptives when they first became available. And so I just would put this statement up from Planned Parenthood that any method of birth control is more effective than no method, and the most effective method is the one the couple will use with the greatest consistency. And if you change birth control to HIV prevention, I think that the statement holds really, really well. And the fact that PrEP is one additional option. It's not for everyone, but there are people for whom this would be the option that they would use, whereas they wouldn't use others. And so it can play potentially a big role in preventing the spread of HIV. PrEP is here. And so this is actually, I was in San Francisco in September, and this was one of the subway stops. That yellow line is the yellow line that you can't cross before you get on the subway. PrEP is here, and so it is being implemented, and so we're going to learn more about it as we go on. And I thought I would close by talking a little bit about continuums of care. Going back to people who have HIV and thinking about antiretrovirals and this goal of getting them virally suppressed. What does it take to get someone virally suppressed? It's a good goal. It's good for their health. It's good for their partner's health. What does it take to get there? They have to be diagnosed, so they have to know their status. They have to be directed to care. They have to stay in care. They have to get the prescription, and then they have to actually take the pills and have them work. And here in the United States, we do a surprisingly poor job of actually getting people to viral suppression. Viral suppression being a low viral load on antiretrovirals. So there's a little bit over 1 million people in the United States currently infected with HIV. This slide is from 2012. This number has ticked up a little bit, but not much since then. So a large number of people living with HIV currently in the United States are not virally suppressed. And we lose people at every step of this cascade. There's 1 in 8 or so who don't know their status. Of those who know their status, not everyone is getting referred to care. Of those referred to care, there's a huge drop-off here in terms of keeping them in care. And then once they're in care, we do pretty well at getting them on antiretrovirals and virally suppressed. But there's a lot of challenges here. And then from the prevention standpoint, the 2/3 that are not represented here, so the other 63% that are not retained in care, that is where 90% of our new infections come from. So people that are in this group are much less likely to pass the virus on than the people that are not in care. And so again, the more that we can do for treatment, the more that we can do to bring people into systems of care, the more that'll give us a handle on prevention. Now, why would someone not be in care? There's a variety of reasons. LA County had a program going on for a few years, a navigation program. Out of that study, these are the reasons why people had barriers to care. They had other life priorities. HIV was not their number one pressing issue. And especially in the early years of an HIV infection, if you're feeling healthy and fine, people have other things that they view as being more important. There's a huge connection to poverty, so there's a lack of money, a lack of transportation. Here in LA, we don't have very good public transportation. And so I've talked to people who have to be on a bus for an hour or 2 hours to get to a doctor's appointment that would be really easy to access if they had access to their own car. Substance abuse is a big one. The stigma around HIV, housing instability, fear that someone would find out that they're positive, not really understanding the need for their care, and having to deal with their immigration status and sort of their residency between countries. And so there's a number of reasons why someone might be out of care. And that's why I've really come to appreciate the work that Foothill AIDS Project does. When I first started working with Foothill AIDS Project, I was expected to be introduced to their doctor and their nurses and their pharmacists and so on, and they actually have none of that. That's actually not what they do. They do the wraparound services. So they do all that other work that has to be done to keep people in systems of care and meet their other needs. Now, FAP doesn't see everyone who has HIV. We see at FAP clients who have HIV and these other life challenges like poverty and homelessness or drug addiction. And so we at FAP do case management, we do housing. We have housing is healthcare is our mantra, and it's often where we start with supportive housing, which then helps us meet their other needs, including their HIV needs. And so our budget, the largest fraction by far, goes towards housing. We do a number of support groups, address as best we can people's mental health needs, deal with their substance abuse, offer food assistance, transportation assistance, and do health education. So these are all these missing pieces that you need in order to be effective in responding to HIV. So I'm incredibly proud to be associated with this agency. Just to wrap it up then, my take-home message is there is no cure for HIV yet, but HIV is treatable right now. HIV is transmissible, but HIV is also preventable. And finally, people living with HIV should be treated with dignity and respect. And with that, I'll close, and we'll be happy to answer any questions people have.
We now have time for questions and answers. If you'd like to ask a question, please raise your hand, and Sarah or I will come to you.
Hey, thanks for your talk. My question is actually pretty simple. How does treatment and fighting HIV differ from the strategies on other STIs?
Well, I think so the question was, how is treating HIV different than dealing with other STIs? I think there's a few differences. One is the stakes are a bit higher in that HIV, we have no cure, we have no way of eliminating it, period. And so if someone acquires HIV, that's a permanent thing. Like they will, until we have the cure, have HIV for the rest of their life. The other thing too with HIV is the stigma associated with it is much stronger. And so talking with people who are HIV positive, they'll talk about the ways that they've been treated at clubs or in their relationships. And there's a lot more stigma associated with that of being HIV positive, as opposed to that having had an episode of syphilis or an episode of chlamydia or an episode of gonorrhea that was treated and has gone away doesn't carry with you in the same way. So I think those are kind of the main ways in which they're different. Obviously, the things that you can do to prevent other sexually transmitted diseases also apply to HIV in terms of behavior modifications, or the use of barriers like condoms, definitely do apply. But I think the stigma is probably the biggest one, which is connected a lot with the history of the disease and also the way that it is a lifetime-long infection. Yeah, great question. Thank you.
Hi, thank you. So you mentioned how the stigma around HIV is a huge problem for some people accessing treatment. Mm-hmm. So how do we go about destigmatizing HIV and AIDS? And how is education involved with that? And what are the steps, I guess, that we can next take to, solve that problem?
Yeah, that's an excellent question. So how do we deal with the stigma? So my personal approach is whenever I get invited to speak, I say yes. And so I've gone to Rotary clubs. I've gone I'm here at the Ath tonight. Yesterday I was at All Saints Church in Pasadena. Whenever I'm given the invitation to speak to any audience, I go and I talk about it. And so I think that's one thing that works really well. Another thing that works really well is that I have friends who are HIV positive who are really committed to this issue, very vocal on this issue, and are very unafraid to stand up and talk to a room of people as someone who is HIV positive. And so I've met some wonderful people who do this work, and they do a great job of putting a human face to it, of making it personal. And I think that helps a lot. I think in general, as a country, we need to do a lot more with sexual health education. And it's part of the problem that we face is that there's a large amount of local control in this. And there are school districts that are very progressive in their approaches to sex ed, and there are school districts that are stuck in the 1950s in terms of their approaches. And so I think that one of my students who took my class on HIV/AIDS last year was a Pitzer student, and she's now in a Watson Fellowship, sort of looking at this really important issue. I wish I had more answers to say than that. Yeah. Other questions?
You mentioned the problem of people skipping doses.
Yes.
And anyone who takes any kind of medication knows that there are times you're going to forget.
Yes.
So how many times what happens if you skip once or for a week? Or in other words, how long does it take before it really has an impact?
Right, that's a great question. And the good news is it's getting better. Like the newer doses, because they're more potent, because they have a longer lifetime in the blood, they're much more forgiving than the old drugs were. In fact, they're looking at protocols now with teenage patients of giving them their weekends off, where you take your drug 5 days a week, and then you get Saturday and Sunday off, and then you go back on. And with the newer drugs that are more potent, that have a longer lifetime in the body, it's possible to start thinking about regimens that way. We'd love to eventually get to a place where we can do something like a long-acting injectable, similar to what's done with contraception, where we could have an injection in the arm that will then last you a month or a few months to get around that problem. To show you just some data, I'm going to see if I can find it here really quick. It doesn't actually take that long, especially with the old drugs. So this was a study done in 2000 where they did a structured interruption as a study. And so at week 0, they stopped the treatment, and then the red line is the viral load, and they followed the viral load to see what would happen. This was before they knew that HAART wasn't curative. They thought it might be. And so for this patient here, for example, after 1 week, there was still a viral load undetectable, which means less than 50. At week 2, there were about 700 copies per mil. Week 3, they were at 10,000. Week 4, they were at 100,000. And so it's pretty consistently, week or 2 leads to full viral rebound. It's certainly an issue. And the worry is that when you do this, and this was like a full stop, what's actually even more worrisome, though, is like, say if you take half the doses, and so then the drug is present, but at a sub-therapeutic level consistently, that then gives a selective advantage to viruses that have mutated to escape that drug, which would then become the dominant strain that prevails in the body. And so that's the way you get resistance. And then you have to switch regimens because that particular drug combination would no longer work for you.
What's the state of the science right now on vaccination?
Great question. It's not my huge area of expertise, and so I'll just tell you a little bit just from an outsider perspective. We're not there yet. We have had a few rather public, big, splashy failures, which were frustrating setbacks. The closest we've come that I'm aware of, and it may have progressed a little bit since then, is a vaccine that was tested in Thailand, the RV144 trial. And this was from a few years ago, and it seemed to be about 30% efficacious. And so it would, compared to placebo, it would reduce the risk of transmission by about 30%. That's not particularly good, and so it's probably not ready for prime time yet. It's a very challenging virus to vaccinate against. For one thing, it literally is a wolf in sheep's clothing. It cloaks itself. Let's see if I can come up with a picture that shows this. I can just find that slide. It coats itself in sheep's clothing. And so this is an image of the envelope protein, which is on it's the outside, it's a spike on the outside of the virus. This is the viral membrane. This is a spike that comes out of the virus. And all of those blue patches there, those are carbohydrates that are derived from the host cell. So they're human synthesized carbohydrates that look like us. And our immune system is programmed to not recognize self. Like there's a very strong bias against not recognizing self, for good reason. That's what leads to autoimmune disease. And so the virus is literally coated with all these human-derived sugars, which then make developing an antibody response very difficult. And this slide is indicating like particular sites where particular antibodies have been discovered that do bind to the envelope and neutralize it. We haven't yet figured out how to deliver a shot, an injection, that would stimulate the body to make these antibodies on their own. I think the hopeful thing is that we've found antibodies that can do it, and there are people even who are looking at giving these antibodies in a passive mode. What we'd love to be able to do is to come up with something that we can inject that would stimulate the body to make antibodies like these, and we're not there yet, to my knowledge. That would be the other great way to stop the HIV epidemic. There are people who think we can do it now with the tools that we have now, just through treatment as prevention. Clearly having a vaccine would accelerate our progress dramatically. And to date, the only infectious diseases that we've been able to eradicate or come close to eradicating it has been through vaccination. We haven't yet been able to treat our way out of an epidemic.
Good question. Oh.
Thank you very much. Can you comment a little bit on how HIV is being transmitted today and whether it's the same as back in the 80s and who is transmitting and just kind of the state of the transmission?
Sure, of course. It varies a lot in terms of which country we're talking about and what region we're talking about. Eastern Europe is driven primarily by injecting drug use. There's unfortunately a very large outbreak going on, a growing outbreak in Eastern Europe that's related to the injection drug use and the heroin epidemic. There's a lot of the countries there that have a very non-progressive approach to injecting drug users, and I think that has real consequences for continuing the spread of HIV in that way. In sub-Saharan Africa, it's primarily through heterosexual contact. Here in the United States, it's the main driver is men who have sex with men. I have some data here on that. So this is United States from 94 to 2006. If we keep it going out another 10 years, the trends look pretty similar. I mean, it's kind of levels out. And so male-to-male sexual contact is the primary one, followed by heterosexual contact, injecting drug use, sort of a mixture of those, and then kind of a miscellaneous other. And this would be blood transfusions, mother to child, things like this. If we break it down by male and female, for males, it's predominantly men who have sex with men, transmission, followed by heterosexual transmission, and then injecting drug use. For females, it's primarily heterosexual contact, and then also injecting drug use. Injecting drug use, we know an intervention that works really well, and that's needle exchange programs. These have been shown to be really effective in stopping the spread, and they also have been shown to either be neutral or perhaps positive in the way that they get people into treatment and then off of drugs. And so actually either, they definitely don't promote drug use in terms of increasing the numbers. They might be neutral and they might actually reduce the number of people who use injecting drugs. So that's one way that we could deal with reducing these numbers here.
Okay, I will ask my question.
Okay.
So I'm wondering if you could talk about the relationship between government policy and the effort to come up with a treatment for HIV/AIDS, and then maybe in particular in light of the election of Donald Trump as president and his proposed cabinet and governing style, what you think that'll mean for the treatment community?
I think as with a lot of things with the new administration, we don't know, and so we can guess, we can speculate. We don't know yet. With regards to injecting drug use, one of the things that Mike Pence did when he was governor of Indiana was he had a ban on needle exchange programs, and there was an epidemic that broke out in Indiana for several months among injecting drug users, and under a lot of pressure, Governor Pence reversed his decision and then allowed a needle exchange program to be put back into a place, and that helped halt that epidemic. And one of the things that has been really important as we think about this is getting people into care. And the Affordable Care Act, while not perfect, one of the things that it did do is get people into care and increase the number of people who are seeing their primary care physician. And HIV testing has become a routine part of primary care medicine. And so the more that we can get people into care, the more likely they're going to be diagnosed and sort of retained in care. The funding that supports HIV service delivery, especially like those wraparound services that I talked about that Foothill AIDS Project delivers, is primarily through a mechanism called the Ryan White Care Act. It's named after a boy, Ryan White, who acquired HIV through a transfusion and died of AIDS. And this is a really, really important funding stream to help this work continue, particularly in thinking about these very aspects and the wraparound services that enable you to get people and keep them in care. And I think there's a lot of concern about the renewal of the Ryan White Care Act funding and the possibility of those funds being diverted towards other for other uses that are not scientifically and evidence-based. So at this point, I can express concern, but I don't have any specifics which lead to any sort of certainty about what's going to happen.
Hello, thank you for coming to speak. What are the costs associated with therapy for ARVs and PrEP?
That's a great question. So it depends where you live. If you're in the United States, it's quite expensive. Something on the order of $13,000 a year, that's because these are brand name drugs and you're paying brand name prices. Generic price, sort of cost of manufacture price, is something more like $400 a year, so something like about $1 per day. And so one of the things that has happened in recent years is through a lot of pressure, pharmaceutical companies have made the drugs available at cost of manufacture, either through direct sales or through enabling generic production in the parts of the world that cannot afford it. So places like sub-Saharan Africa, the aid agencies that are purchasing drugs are paying something like $400 a year to care for people.
With all the progress that's been made, can you say anything about the number of deaths per year due to AIDS, both in the U.S. and worldwide?
So, let's see, I'm trying to think if I have the exact numbers. Give me just one quick second. I might be able to pull up the slide. If I can't, I'll do my best to represent it. Yeah, I don't think I have the slide here. So in the United States, I'll come back to this one. So this is the death rate per 100,000 in the population, actually of all sorts of causes. Sort of interesting to see what people are, and this is for a particular age group, 25 to 44. Something like 4 per 100,000 on an annual basis, and so it's relatively low. Globally, the death rate from HIV, the mortality rate, is something on the order of about 1.9 million per year, which is an extraordinarily high number, way too high. It's down from a peak of about 2.5 million, and so we've made a lot of progress. And so, 2.5 going from 2.5 million down to 1.9 million feels like great progress. 1.9 million is still a really, really large number, and so I think we still have a lot of progress to make. Globally, there's something like 33 million people living with HIV, of whom about 12 million are in treatment.
Thanks, Karl. Would you comment on World AIDS Day celebration on Thursday?
I would be delighted to, yes.
Good, thank you.
Yeah, so this is my dear friend Georgianne Andrews, also associated with the Fiddle AIDS Project. Today's talk was sort of part of the lead up to World AIDS Day, which is on December 1st. It's been celebrated on December 1st for a number of years now, and to mark the occasion, Claremont, UCC, the church at the corner of Harrison and Harvard, has an annual World AIDS Day service, and this occurs at 7 p.m. on Thursday evening. And following the service, there is a candlelight walk that'll actually start from the church there, through the colleges in front of the library and down to an art museum at Pomona, and then back to the church for a reception. It's a beautiful event. It's a time that many people who lost loved ones to HIV come to remember the ones that they've lost. It's also a time for people who are living with HIV to gather and sort of in solidarity. It's a beautiful event. Again, that's Thursday at 7 p.m. at the Claremont United Church of Christ.
What kind of side effects are there to the drugs?
That's a great question. And so, so what sort of side effects do the antiretrovirals have? Drugs, the side effects were many, and they were quite severe. And the things like, highly elevated risk for cardiovascular disease, an imbalance in your lipids, and we've moved away from, and we think that a lot of those side effects were due to the protease inhibitors class of drugs, which is part of why we've moved away from that class. The newer drugs, and so like let's say Truvada, for example, which is the drug that's used in PrEP, the biggest toxicities are renal toxicity, and so you can monitor that by doing blood tests and watching out for renal function. What we know is that the side effects are there. The drugs, though, have fewer side effects than the virus. And so there was a study that was done in stopped in 2006. And what they did with the study is they tried to give less drug. And so they would have people who were stably virally suppressed, and they would take them off their medicine to spare them the side effects of the drug, and they would put them back on when their CD4 count fell. That was what one group did, and then the other group, and this was done by random assignment, just stayed on their meds continuously. And so it was sort of a competition to see, all right, if you give less drug, so fewer side effects of the drug, but more virus replication, is that better or worse than more drug, which is more side effects but less virus? And so it was a randomized clinical trial, the SMART trial, Strategies for Management of Antiretroviral Therapy. And they actually had to stop it early. The Data Safety Monitoring Board halted it because the results were so clear they couldn't keep on going. And so the group that had the treatment interruptions, that had less drugs, so fewer side effects from drugs, they had more HIV-associated problems. And I think everyone kind of anticipated that. But the hope was that by giving less drug, there would be fewer of these side effects. But actually what they saw instead, and it was sort of surprising, and especially how dramatically strong the effect was, the people that took less drug compared to those who took more drug, the people who took less drug had more heart disease. Heart disease was known to be a major side effect. More kidney disease, which was also known to be a side effect. More liver disease, also a side effect. And just more morbidity and mortality from all causes. And so on essentially every measure they looked at, having less drug actually made the person sicker. That the virus itself is toxic. The virus itself damages multiple organs, not just the immune system, but multiple organs. And so because of this, this was part of this was one of the studies that led to the recommendation of this universal prescription of the antiretrovirals, that regardless of CD4 count, regardless of whether the person is sick or healthy, the antiretrovirals, because the virus does more harm than the drugs do.
Fortunately, that's all the time we'll have for questions tonight. Please join me in thanking Professor Haushalter.